Good afternoon. So my name is Rayan Chikhi from Institut Pasteur. I will give a talk about recent progress towards petabase-scale genomics. Actually, I have a longer introduction. So your instructor is not only jet-lagged. I just flew in yesterday from Paris, so apologies if I seem more tired than I usually am. And apart from jet lag, I'm also a PI in bioinformatics algorithms at Institut Pasteur. We are a computer science lab inside of a microbiology lab. There's some parts of my CV here. And my previous research focused on genome assembly, methods based on k-mers, metagenomics, and large-scale computations. These are some pictures of species I contributed to assemble. And you can find me on Twitter. And what happened? What happened? All right, let's keep going. And this is one of my pitch. So this talk, wait, the volume has been done. Can you still hear me? Perfect. A little bit. So something happened to the volume of the microphone. I guess I will have to be closer. So this talk will be about two parts. One part is recent large-scale genomics. And part two will be about a story for a recent project I contributed to called CNRS. And it's about a very large-scale analysis of genome sequences. So let's start with part one. So my first example of a large-scale sequencing data analysis is on plants, plant genomics. You might remember, at least for the faculty, a talk from Mike Chatz at UCLA 2019, where he was telling us about an analysis of tomato sequencing. So they sequence 100 tomato genomes. Each tomato genome is a 1 gigabyte, gigabases in length. They discovered lots of structural variants. And this is one of the most comprehensive pangenomic structural variant survey so far for a plant. There is a nice story about the clue gene. I don't know if Mike talked about it in 2019. Anyone remembers? I really like it, so I'll still mention it briefly. So this is a gene, which is, the name is the inverse of Hulk. And in fact, it is responsible for the size of the tomato. And before, scientists thought that a SNP inside this gene affected the size. But after this study, they realized it's not a SNP. It's actually a copy number variant. When you have more copies of clue, the tomato is bigger. If you have less copies, it's smaller. So I would argue this is the least well-named gene ever because it should be named Hulk and not clue. The more you have it in number of copies, the bigger the tomato is. So not only tomato analysis, but also humans have been benefiting from larger scale sequencing. So for instance, this paper, which came out recently, provides the sequences for more than 100,000 whole genome samples, not just SNPs, but whole genomes from biobank. And they did a pretty extensive analysis of SNPs, so 500 million SNPs, 7% of all possible SNPs. Lots of indels, lots of structural variants. In terms of methods, they extensively used a software called GraphTyper. And they compared it against GATK, and they found better performance for SNP calling. And of course, in this paper, they also perform associations for rare variants with large effects. So I suppose since this is CGSI with lots of emphasis on human population genomics, you might know about this analysis, but I just wanted to highlight that this is a really large-scale analysis, not just on SNPs, but on many other signals. And finally, last example is not just about large amounts of data analysis, but fast, critical medical analysis. So they performed sequencing to diagnostics in less than 6 hours. And what I want to talk about is not exactly the results, but how in terms of infrastructure. So they used the cloud to distribute calculation in multiple computers. So for instance, sequencing data is provided here. And then base calling is done on multiple computers, alignment and SV calling the same, and finally annotation. So this is a nice example of not just critical algorithm development, but also big data analysis in a sense that they needed all of this computing power for fast analysis. So all these analyses are being carried because more and more data is becoming available. So for instance, sequences, they provide, let's say, 1/3 of terabytes per run. Your laptop could reasonably contain more or less 3 runs. A recent large-scale analysis of DNA and RNA in oceans was performed by the Tara Ocean Project, and they generated 60 terabytes of sequencing data. And even that is really small compared to the amount of all public DNA sequencing data that exists, which is roughly 30 petabytes nowadays. All of that is also very small compared to Google servers and, for instance, YouTube. But this gives you a sense, a scale of genomics nowadays. So at this moment in time, the biggest data we can manipulate is roughly at the exa scale. So what I mean by exa is really if you pull together all data from YouTube, genomics, astronomy, and so on, that's the scale. We are more used to seeing, for instance, gigabytes, terabytes, and for a single project, maybe petabytes. And actually, the next step in time, where we are not yet there, would be zettabytes, but we haven't seen this amount of data so far. This is just to recall the units. Can you still hear me well? Because I don't get any feedback now. Should I speak louder? Yeah. OK. All right, so in terms of data analysis, all of this has been foretold by a researcher called Titus Brown at UC Davis, who's been giving talks since 2017 in metagenomics, and he contributed to many k-mersed-based methods and analyses. So my argument now is that not only we are used to big data analysis, but this is being replaced by bigger data analysis. So a basic element is that one computer is not enough to carry all of these analyses. We need to use what computer scientists know very well as parallelism, which considers, well, many computers to perform a single analysis. So you start from data as input. You somehow distribute it to many cores or computers or something that can compute, and then you have the output of the analysis. Unfortunately, this cannot scale indefinitely because of an annoying law that is known as Amdahl's law. It says essentially that depending on how much of your computation can be parallelized, the speedup is capped to a certain percentage. For instance, if, let's say, half of your task has to be done sequentially, then you cannot do better than 2x speedup, no matter how many CPUs or computers you run it on. So you might say half is perhaps unrealistic, but let's say 5% of your task cannot be optimized and has to be performed sequentially, then your speedup is still capped at 20. And nowadays, servers have way more than 20 cores. So this is really critical for performing large-scale analysis. And I wanted to provide a small exercise for the audience to connect the dots from tasks to the left and times to the right. And I'm giving you some helping metrics here. So for instance, how much time does it take to read a small file on disk, to access data on memory, to open a web page in Australia and align 1 million reads? If somebody knows, can just tell me the results in terms of numbers. Is it 1, 2, 3, 4, 1, 3, 2, 4, and so on? Any idea? I know CGSI doesn't have any practical sessions, but I'm still trying to involve you in some thinking. Who says 1, 2, 3, 4? No one. Any candidate?
2, 1, 3, 4.
There seems to be 2, 1, 3, 4 being said. Who agrees with that? A good 20 people, or actually 30 people. So yeah, that's 2 and 3, 4. So either you know it or you don't, I guess. So I guess accessing data in memory is the fastest. Then comes a disk. Then web page opening is limited by the speed of light. And especially if you don't live in Australia, light has to travel somehow between your location, and that usually takes in the order of milliseconds. And finally, handling a million reads is the slowest operation due to, well, the complexity of alignment algorithms. And I think this metric was given for BWA. I want to do a brief aside on programming languages, which is a highly subjective take, of course. But my main message of this slide is not that PHP or MATLAB is bad, not only that, but it really doesn't matter what programming language you use. For this talk, if you consider parallelism, the speed up or speed down you might get of using Python versus C++ is completely irrelevant to the volume of data being processed, for instance. So another question to you is, what are the largest parallel computers that you can access as a genomics researcher? You can give me many types. I'm happy to take multiple answers. Like if you have a big analysis to do, what do you use? AWS, Google. These are good answers. There's more possibilities. But you can also use your own university cloud. It is a parallel computer. But in fact, there are many solutions nowadays. You don't really think as a graphics card as a parallel computer, but it really is one. There are some dedicated infrastructures from FPGA. So for instance, Dragon at Illumina provides a fast way to map reads and do variant calling. Local cluster, national cluster. I put quantum computing and DNA computing in there, even though they are not ready yet for production, but hopefully they will at some point in next decades. In terms of public resources, you might not think of Galaxy as a powerful parallel computing architecture, but in fact it is. And then there is Envol and TerraBio, which are specialized for certain types of analysis. And finally, all of this can be orchestrated by some execution engines, such as Nextflow and SnakeMake. So I want to do one more sound test, because I think I try to speak as loud as I can, but I believe the microphone is not really picking up my voice. So is it fine on the back, or still not great? Fine? OK, thank you. Yeah, is this better? I guess I don't even need to read this slide. It's pretty self-explanatory. Oh, I keep this one? All right. Thank you. All right, 2 mics. Is it better now, or is it the same? Same? Better. OK, great. Let's keep it this way. Thank you. OK, so it's actually, from this slide, it's hard to get a picture of a cloud publicly, because usually they don't distribute it publicly. So this is one from close to my city in France. But if you look for AWS or Google Cloud pictures, they're all fake on the internet. They're not the real ones. Because I guess this is like a trademark secret or something. On the cloud, you can get different instance types, which means different types of computer optimized for different tasks. In genomics, we are often interested in one of those 3. So general purpose, or many CPUs, or much RAM. Accelerated computing would be for machine learning, and storage optimized is usually databases. So they have different number of CPUs, different network speed, and different cost, of course. And typically in 2022, this is the cost that you can expect to pay for cloud computing. So for instance, for something looking like your laptop, it costs not so much. It looks cheap, but in fact it is not, because it's roughly $100 a month. So the laptop that you have has a value of $100 a month of cloud. So that's pretty expensive if you keep adding up laptops. However, for beefy analysis, then it gets more practical for a week, for instance. So what it looks like once you enter the administration portal of, let's say, AWS, you can start, stop instances. And it's easy to make mistakes. If you forget to stop an instance, the build keeps running and so on. And at this point, I will move to part 2, but I'm happy to take questions about the first part. Oh, well, the microphone has changed. Is it better now? Ah, I guess I needed to go to part 2. Huh? So what happened? Any question about so far? No? Because you didn't hear anything, did you? All right, I guess now this is the interesting part because you can hear about it. So I'm going to tell the story about the petabase-scale discovery of viruses that we did 2 years ago. We analyzed all available RNA sequencing data and discovered 10 times more viral species than previously known, including some new coronavirus species. The project is called CNRS. I contributed to it, but I should mention that the masterminds of this work were Artem Babayan and Robert Edgar. The context of that work is unsurprisingly viral surveillance in the age of pandemics. So, short story long, long story short, I suppose. We're here at the pandemic stage, and we would like to avoid the pandemics by doing pathogen spillover surveillance as much as possible. And this is important because we all know that SARS-CoV-2 has circulated and keeps circulating among animals. We've known that well, after the pandemic has happened, but it keeps going. So tigers, dogs, and minks keep being infected, and we wish to prevent such infections spilling over back to humans with new variants. To do that, it's important to sequence and monitor the spread of coronaviruses, and in fact, any viruses. And there are 2 types of sequencing. One which is targeted to SARS-CoV-2, especially in humans, so this is next strain and other efforts. And these are other types of sequencing which target not viruses, but environments. For instance, the Tara Ocean project just sequences everything in the ocean. In both cases, data is being deposited to a repository called SRA. How many of you have accessed this repository before? A large, more or less a majority, right? So what you can do is search by metadata and download reads. Essentially, you cannot search by sequence. If you could, it would be helpful for the community, but it's too difficult in terms of algorithms. There is no blast for SRA. I wish there was. I mean, we're working on it. The database is big, so SRA is now 30 petabases in length. So if you remember my units slides, it's a lot of data, 30 millions of gigabytes. And it keeps doubling roughly every 2 years. So in 2022, it's 30, but in 2024, it will be 60. And we can't keep up with this doubling. So whenever I think about the SRA, I more or less think about the data crypt, where the data sleeps undisturbed, never accessed, and well, it's a shame, right? So we would like to change that. And in particular, if we focus on RNA viruses, we should focus on RNA sequencing. And in the SRA, there are 5 million samples as of 2020, which represent 10 petabases. If you were to download all of this data, not even analyze, just download, how many years would it take? Any estimate? Too many is a good number, but what about more precise? 1,000 would be a reasonable estimate. Yes. 300 would be an even better estimate because of the number I was giving you here. And in fact, guess what? Google can even give you the answer if you just ask it nicely in a proper search term. So it's 300 years. It looks unmanageable, but thankfully we have parallel processing. So the CNRS project managed to do a cloud analysis of all of RNA-seq data, so the one that would take 300 years to download. Because something happened in 2020, this data has been mirrored to AWS, to Amazon, which means that we can access it much faster than the speed I gave in the previous slide. So in CNRS, we did two analyses. The first one is a nucleotide alignment of all the RNA-seq versus all RNA viral genomes known. And through this analysis, we discovered some unknown coronavirus species. Then we aligned all RNA-seq to all known RNA viral marker genes. And through this, we discovered new RNA viral species, quite a lot. You might ask, why two analyses, not just one? It's because the first one wasn't sensitive enough. If you do nucleotide alignment, you miss high divergence sequences. So I'll briefly talk about analysis 1, where we did nucleotide alignment using BOTI2 of all reads to all known viral reference sequences. And we found that SRI contained 55,000 samples that contain some coronavirus. And so what's interesting here is, I guess, the cloud architecture that we used. So we download data from Amazon and then download and alignment to lots and lots of computers. I think at the peak, it was running at 20,000 CPUs at the same time. And then all alignments are merged, stored, and everything is stored on a public dataset, on a public repository. So it was aggressively cost optimized, specifically to avoid the problems of Amdahl's law, in order to avoid bottlenecks. And I should mention that all the code is open source. In terms of algorithmics, there is no news, so no news in particular. So the alignments were done using BOTI2. Download and merging is pretty standard. But I guess the novelty here is that for the first time, we are able to analyze 1 million samples in a day. So that's really fast. At a cost which is half a penny per library. And you might wonder how much such an analysis costs, and the cost breakdown is here. No wonder downloading and alignment is really what takes the most time and money. So this enables some results, and since this is CGSI, yeah. I think total, so I should mention that we did some test runs, but for a single pass over the whole SRA, it's in the order of $10,000, which is expensive for a grad student, but not so expensive for a small grant. So it's within reach. So this enables to find some interesting things. So for instance, we analyze all of these data sets. Every time there's a dot, there's one geolocalized sample, so it's all around the world. And we perform again this analysis another time, because we realized the first one was not sensitive enough. So we download again all the reads and align this time using Diamond 2, which translates into 6 different frames, and then compares against all the known versions of a certain marker gene I will talk about, of all RNA viruses, and finally we get some BAM files. So in particular, the marker genes were the 15,000 known RNA viral species RDRP gene. It's useful to think of RDRP as the 16S barcode of RNA viruses. And in fact, it's present in every species in polio, coronavirus, and other families. So what Artem and Robert did was extract particular domains of this RDRP gene, which were highly conserved across all species. And this is what they call a PAM print. And they wrote a software to detect PAM print from reads. So it's pretty short. It's 100 amino acids total for a PAM print. And then once this barcode has been defined, one can define virus species based on it using amino acid identity thresholds. Then, once we get these RDRP genes, it's just possible to microassemble or just assemble the reads which matched the RDRP gene within each sample. And I did that using existing tools, Spades assembler, and GNU parallel. So just a quick word about results. So the left part of the figure shows distribution of number of libraries, actually petabases inside libraries. So surprisingly, SRA contains lots of humans and mouse. Also lots of, we took also some WGS, so DNA as controls. And we found many, many more viral species within different environments. So unsurprisingly, viromes, environmental samples, metagenomes, but also some within human and mouse. So they are from different origins. And I will skip explanation for that one, but it shows essentially novelty using sequence identity to known RDRPs. So it's possible to access all of this data. And in fact, we didn't analyze it all, but we made it public on this website with some browser to allow other researchers who are specialists in viruses to study more specifically one family. I'll briefly mention one thing about coronaviruses, is that the analysis 1 enabled us to discover 9 more species of coronaviruses. And these are all in fishes. So initially there was one nidovirus, which is a family above coronavirus, known in Pacific salmon. And now we found more in axolotl, fogo fish, seahorse, I think. And we call it group E. So there is alpha cov, beta, gamma, and so on, delta. And group E is more distant, but it's still a coronavirus. And there was one particularity about these viruses, is that in all the assemblies, they were missing the spike gene, which is pretty surprising because all the viruses that we know have spike. So that was a big surprise, but we went back to the data and tried to assemble it. This was joint work with Anton Korobenikov, and we actually found the spike gene in another context. And consistently, we were never able to computationally close the gap between the left part of the virus and the right part. And in fact, in the Pacific salmon nidovirus, the gap did not exist, but in the sequence there were ends. So this suggests that those viruses are segmented into 2 different molecules, which would rewrite the definition of what a coronavirus is. All right, so for this analysis, we perform metagenome assembly. And usually when you do that for a sample, it consists of reconstructing all the genomes of all viruses and bacterial species in a sample, and this is computationally intensive. For the first analysis, we were just focusing on recovering coronavirus genomes. But this was also computationally intensive because it required to reconstruct everything and just filter at the end. For second analysis, we only had reads mapping to RDRP. And this is computationally easier because the volume of data is much smaller. For analysis 1, in fact, the Spades team has written a specific assembler called Corona Spades to better assemble, to better scaffold across genes of viruses. Just to give you a short glimpse of what it looks like to perform large-scale genome assembly or large-scale computation in general. So I was using something called AWS batch, where you can automatically launch many instances. And so for instance, in this job, there were 100 instances running at the same time. Please disregard the number of failed jobs, but otherwise it worked well. For analysis 2, only a single instance was used, but a pretty large one. So in terms of what's next, there were there are now 10 to the power of 5 our species now, but we know that there are 10 to the power of 8 species left to discover. So that's a lot of analysis to be done yet. Also, we didn't look at DNA viruses at all. It would be nice also to detect lower homology with non-RDRPs. However, bowtie and diamond don't support it. But there is interesting research from Google Research Paris, which looked at deep learning for lower homology alignment. And finally, one thing which is close to my interest is to be able to index the whole SRA. So it's nearly feasible with k-mers already, but it would only support exact search. And it doesn't exist also, that's the problem. All right, so in summary, we discovered 100,000 new RNA viral species through this project, a new group of coronaviruses. In the paper, there are also analyses for other families, such as very compressed viruses, and delta viruses, huge phages, and so on. All of the data is accessible on GitHub. There's a lot of alignments and assemblies to analyze yet. More details are in the paper, on GitHub, and on the Slack. And I will mention that this was the result of a worldwide collaboration that started from a hackathon. It's quite unusual. So this is not really an academic project, but more hackathon initiated by Artem, which gathered anyone who was interested in participating. And in fact, what happened is that at the end, we were all first author of the paper, which is also pretty unusual. And in fact, we never met in real life. So it was all by Slack and Zoom. And I will finish with some slides from Martin, which I really like. So in the hundred of years that we've been doing microbiology, we've known 15,000 RNA viruses species. And with less than 1 year of computational bioinformatics analysis, we discovered 10 times more. But this is really small compared to the size of virome that exists. Okay, so in summary, lots of genomic data, many great analyses could be made, and the cloud helps at a larger scale. So some credits to people which I don't have time to name, but at least display here. I mentioned that some people from my group are here at CGSI. Feel free to talk to them. And myself, I'll be here all week 1 and some days of week 2. Actually, I have a second talk at the end of week 2, which will be about introduction to pan-genome graphs to those of you who will be here. I plan to do some live demonstrations, cover some various recent methods. I could also talk about human genome assembly very quickly, but I didn't plan to. So if you'd like to know more about it, let me know. Or convince me to talk about it on week 2. All right, so thank you much for your attention.
Next week will be on the recording, so for those of you that aren't here.
Oh, good to know. Also, I hear that it's hard to hear the questions when you ask them, even from the front rows. So is it possible to use the microphones or something? Yeah.
Okay, it's not working.
Okay.
So 3 years ago, you talked about k-mers and minimizers and all of these advanced algorithms in order to make it computable. And I'm surprised to see that you're using like both.
Yeah. Okay, so the question was, I'm repeating it for the recording, so why, 3 years ago I was talking about k-mers and minimizers, so why didn't we use that for this analysis? So you would be pleased to know that in fact bowtie is using k-mers under the hood, but it's not indexed in the right way. So short answer is that no one has constructed a public index of the SRA in terms of k-mers yet. If we had one, we could do exactly what I was talking about three years ago.
And it would be more efficient, right?
It would be way more efficient. Instead of wasting all of this computing power by downloading all the reads from Amazon, we could just do one little query on the index of a certain set of k-mers, and it would give us a good start at least. It would be way more efficient, but we are not there yet, and that's a research goal for the whole community. Yeah?
Yeah, I had a question about this new genus of coronavirus you discovered,
Right.
the segmented one. So I was curious if, like the second part, like after ORF1A and 1B, is there like a later sequence, like at the beginning? Like, did you assemble these?
Right, so every time you see a line here, it's a quantic that we assembled. So
Awesome. Okay.
I think the first group of lines correspond to the accelerometer. I didn't write it, but we had a lot of accelerometer samples, and consistently the gap happened at the same location, which is quite unlikely given that they are from different labs, different samples, and so on. And it's also consistent with other species.
That's awesome.
So yeah, different quantics. We didn't do any molecular validation, but that's the hypothesis right now. We looked at the assembly graph, and it's really not possible to link those two quantics. Yeah. Yeah.
So we spoke about this database RDRP,
Right.
and can you a little bit elaborate more? And does it have any connection to the ribosomal database, the RDP? You know that?
I don't know that one.
Okay, but can you expand a little bit?
Yeah, sure. So the question was expanding about RDRP database.
Yeah.
So you can extract the RDRP genes from all known RNA viruses and constitute a database of all the versions known of that gene, which is what Artem and Robert did. So there is somewhere on GitHub a FASTA file containing all the known RDRPs to date, and they in fact extracted certain domains from that protein, domains A, B, and C.
Okay.
And from these domains, they extracted motifs. And from these motifs, they designed a way to recognize for any new sequence whether it looks like an RDRP or not. So together with this software and database together, you can run it on any library. And then it will tell you, okay, this library is likely to contain a certain set of RNA viruses. But then the analysis doesn't stop here. You need to reconstruct the whole RDRP gene, and this is done using assembly. Yeah. Thanks. Yeah.
So this is a known thing that there are these 3 regions in the RDRP of every RNA virus that are conserved across that particular species.
Okay, the question was whether this was known, that motifs A, B, and C were conserved. Well, before this work, no. This is really Artem and Robert, which put up a preprint where they argue that this is conserved. It was known that RDRP was present as a whole gene in RNA viruses, but it wasn't known that these 3 motifs were highly conserved until they did this analysis.
Can I also ask one?
Yeah.
For the segmented coronaviruses that you discovered, do you have any idea how they are, how those genes are transcribed when they infect the whole cell? Do you have any idea about that?
Question is how the genes are transcribed when those segmented coronavirus infect cells. Well, no, this is outside of my knowledge, unfortunately. Yeah.
Can you also explain what scientific questions are more suitable to be answered by analysis 1 versus 2?
Yeah, okay. So question is, what questions can we answer best with analysis 1, analysis 2? So as you can see, these are very different queries being made to 2 different databases. One is entire genomes with less sensitive alignment, but more specific. And this is just genes with more sensitive alignment. So I think we really pushed the potential of analysis 2 as far as we could in terms of discovering chunks of RDRP using translated alignment, but it could be pushed further using different types of aligners which maybe don't exist yet. For the first one, I think we didn't exactly extract all the possible scientific results we could from this analysis, because, for instance, now we have, actually even for analysis 2, but we focused on discovering new coronavirus species because that was really the angle of the project at the beginning. But we really left aside all the other RNA viral species. And we do have lots and lots of BAM files from both analyses, and in fact the first one, which tell us that some libraries contain new variants, new strains of other RNA viruses, and we haven't analyzed them. Yeah, thanks for the question. I realized I was way faster, so I still have 1 minute left. But I'm happy to make time for the next talk if there are no more questions.
Okay.
Thank you for your attention.
Better, we actually have a 30-minute break now.
Oh, do we?
The best thing about the CGSI schedule in the afternoon is that there's a break after every talk.
Sweet. All right, so
Fantastic.
thanks again.